Um, so thank you, Sangeetha, for accepting our invitation, and a very warm welcome to everyone on behalf of the Caucus for Women in Statistics and Data Science. I'm also going to take this opportunity to let all of you know that currently we have renamed ourselves from Caucus for Women in Statistics to Caucus for Women in Statistics and Data Science. So a very warm welcome to all of you to our first fall webinar for this international webinar series, and possibly our first uh, industry webinar since its inception or resumption in 2020. I wholeheartedly thank Sangeetha for accepting our invitation. She is currently the manager of Translational Biomarker Statistics in Pfizer, India, within the India-Philippines Global Biometrics and Data Management. She has more than 8 years of experience in the pharmaceutical sector. And before joining Pfizer, she has worked in Z4 R&D solutions, and she has worked on more than 50 real-life studies across different phases and different regulatory submissions. She's a postgraduate of statistics from PST College of Arts and Science. A very warm welcome to all of you, and I cannot thank her enough to take time out of her busy schedule to accept our invitation for the webinar. Without much further delay, let's get started. And I would request, as some housekeeping rules, I would request all of you to keep yourself muted. If you have any questions during the talk, you may write it down on our chat box. And anyways, we will be having some 10 to 15 minutes of Q&A session during once the talk is over. So thank you all, and let's get started with Sangeetha.
Yeah, thanks, Sangeetha, and hi everyone. Good afternoon, good morning, depending on wherever you are logging from. Thank you for the warm introduction, Sangeetha. That was really nice. Let me just share my screen. Yeah. Yeah, are you able to see my screen? Okay.
Yes, Sangeetha.
Yeah. Great. So when Sangeetha reached out for Caucus for Women in Statistics, I was really intrigued because I'm also work closely with gender equality social works, but to be able to do that in the workspace and be part of this, I was really honored to be a part of it, and I'm really happy that I'm here with all of you, and thank you for joining. I understand that we all come from different industries and academics and everything. So what we do in pharma, what kind of statistical applications that we use is something that I wanted to share with you. This is I mean, getting into this would be a huge topic, but I just wanted to be able to give an overview and glimpse of it so that you you can probably understand what it is and how it's going to what is going to be there. So you can probably research further on your own, or get to be part of discussions like this more often. Yeah, so before we begin, I just wanted to share this disclaimer. So this presentation is based on only on all the information that is publicly available. It does not include any of the Pfizer owned products or Pfizer approaches. And the views and opinions in the presentation are strictly of the speakers, and does not endorse Pfizer's values. And these slides are intended for educational purposes, and not for the distribution outside the internet purpose. And yeah, the content of the slide is accurate to the best of presenters knowledge and at the time of production. So before we begin, I understand that we are all from various backgrounds. So let me just quickly share this poll, and if we can answer this, it would be really helpful for me to be able to understand, like the audience that we are uh, having today. Are you able to open the poll, open the link?
Yes.
Yeah, if you're able to open the link, you could just type in this code. I'll also post this in the chat. So you can answer to any of the options that are here. Basically, I just want to understand the discipline that you're from, so that we can. Okay. We have people from academia. We have people from pharma. My Pfizer colleagues are here. Thanks for joining in. Okay. Yeah, we mostly have academia, statistics, non-pharma, and statistics pharma. Thank you. Okay. Starting with biostatistics in clinical trials. So what are clinical trials? Clinical trials, we at least post COVID, we all have a good understanding, or at least we read about a lot, watched a lot of YouTube videos, like talk about clinical trials, how things are, medications or devices being tested and all that, especially with the vaccine, how the vaccine was created. We even read about all the how it was manufactured, whether it's mRNA vaccine, all these things. We read about it during COVID. And for me personally, before COVID, it was very difficult for me to explain what I actually do with my friends and family. But after COVID, it sort of became easier. But yeah, so clinical trials are research studies that test medical, surgical, behavioral intervention in people. So this is not just limited to drugs. All the medical devices, the diets, and any kind of cosmetic products, everything go through trials. But all the clinical or medical products that are in the market would have gone through clinical trials with different phases, etc. So clinical trial basically has is a long process. It takes at least 13 to 15 years for one product to be from innovation to till it gets marketed. It approximately takes 13 to 15 years. So we are so before doing clinical trials, we sort of do a preclinical trial which goes on for years and years. If an approach or molecule seems promising, they'll test in animals and see how it is affecting the animal's body, how harmful the drug is and everything. And then only when it shows to be promising in animal studies, only when we have enough data that this could be safe for human consumption, they will move to human trials. And even then, it's there are uh, lot of other factors that are involved, and we need to check what would be the right dose, whether the dose is safe, how efficient it is, and all that. And that takes another a few years for it to be completely ready for marketing. uh, yeah. So why do we need clinical trials? uh, we have all these questions, right? In anything related to healthcare, we can use clinical trials or clinical studies to sort of bring in a hypothesis or bring in an understanding of it. So basically, if we are creating a new drug, how the drug works, whether how safe it is, and how good the drug is. And then so there are drugs that are in different forms, like you would have seen drugs that are the same drug that would be available, say paracetamol, that is there, there is tablets, but it's also available as syrup. So like that, if so there are needs that we have as human beings, right? So like in other industries, pharma also works based on the human population needs. But the thing is, this is with respect to saving people's lives. Sometimes it's as simple as like having a cosmetic product, but sometimes it's like a cancer drug. So any question that we have related to human body and the diseases and everything, we need clinical trials to verify the compound that we are putting into the drug, whether how it is working, how it's helping, how it's not helping, all that. And to know that, we need clinical trials. So these are the principles that clinical trials are based on. Ethics, so again, we are testing on humans, right? As human beings, we all have our own rights. So how ethically we are conducting a trial, if we are doing a certain procedure, if we are adding even one additional dose, we need to justify why we are doing what we are doing ethically, whether it's right or not, all those things. So in clinical trial, we will have an ethics committee that comprises of people from various disciplines, and they will evaluate the study protocol to see whether it's ethically okay to move forward with this procedure or not. So again, scientific validity and integrity, obviously, and then medical relevance. So this is one of my favorite things to talk about, because as statisticians, we talk about statistical significance, we look at data as data. But when it comes to clinical trials, the data is not just rows and columns, right? This is somebody's actual data. The numbers that I'm looking at now is from somebody who have a disease and who is in a clinic, who's giving out those measurements, giving out those samples. So I am reading this person's information, right? So I cannot just look at it as rows and columns, and I can probably do multiple models and everything. We need to see how relevant it is medically also. See, I can get like statistically significant results, but whether clinically also it makes sense or not, it's something we need to understand. And this is just a beautiful journey of clinicians and statisticians coming together to get this research work done. And yeah, regulatory and medical legal issues, and then costs. Yeah, so this clinical research also involves a lot of cost and lot of regulatory requirements. So we have regulatories in different countries. India, we have DCGI, and in the US we have FDA, and each have their own set of regulatory requirements. And also they have some common ground where they like talk about common problems, common guidelines and everything. And when we talk about cost, like it's millions and millions of investment that we put into a drug. So we need to ensure that we are not like overspending. At the same time, we are not compromising on the quality or safety just to cut costs. So how, optimizing the cost is also like one of the uh, principles in conducting clinical trials. So moving forward, so like I said, clinical trials goes on for like years and years. So in preclinical, which is during the animal study, this could go on for any number of years, because we may have multiple compounds, we may have multiple molecules that we want to test it out, but it may all not be successful. Very few would be, promising, and then they will properly test those on animals and then make sure that it can pass through, human trials. So before that, we need to submit all the test results from the animal studies to the the regulatory body, and then get it approved for mm, human trials. So even in humans, like if I'm doing a drug for migraine, I cannot just, just because I'm doing a human trial, I cannot just go and give it to people who have migraine, right? So I need to first ensure that whether the drug is for the first time is being, like put into humans. So we need to see whether this drug is safe for human consumption first, right? So that's where we will do a phase 1 trial. We have multiple phases. In phase 1, our primary objective is to check whether this drug is safe for human consumption or not. And then the second question would be, what would be the safest dosage possible? So what how they will do is, they'll have, so from clinical trials we have some data on what are all the, possible safe dosages that could be. So they'll increase the doses, like that is called a, a SAD and MAD, which is like a dose escalation models, single ascending dose, multiple ascending dose. So they'll first give the lowest dose to one person, and then check if there are any adverse reactions happening. And if it is proven to be safe, they will, take two more people, and then give them the next, highest dose. If I'm testing with 10 mg, and then I'll probably give 20 mg, and then see the maximum tolerable dose, right? Say if I'm starting to have more adverse events at 100 mg, I'll just stop with probably 50 mg or 80 mg, whichever was the previous safest dose. So that's what they'll do in phase 1, which usually have very less subjects. It says 20 to 100, but usually, like 20 to 50 and not more than that. Um, so they'll do multiple ascending dose testing and see how safe and what is the maximum tolerable dose, right? So moving forward to phase 2, so all these are healthy people in phase 1, because like again, like I'm giving this drug for the first time in humans. So if a person is already have a disease, if the person is already weak, how can they withstand if there are any adverse reactions? So the first, they'll just check with healthy volunteers who can see, if they can withstand the safe dosages or not. And then the second phase would be uh, a little, like bigger trial, would have hundreds of people, and then it will have people who have the disease that we are interested in testing for. Say if I'm doing a trial for migraine, or if I'm doing a trial for arthritis, I'll recruit people who have, arthritis, and then I'll test the same dose that we have fixed in the phase 1 with the phase 2 subjects who have the disease. So again, safety is our top priority here, because this works for a healthy population, but we need to see if it still works for people who have the condition. So we need to check that, and also with the people who have that condition, how efficient the drug is. We need to check the efficacy of the drug, how well it works in these set of people, and like how it improves their life. So that will be the phase 2 trial, to make sure that the drug is safe with the people who have the condition, and also to check the efficiency of the drug. So this won't be a short trial. This will be a little, like bigger trial than phase 1. So because we are also evaluating efficacy here. And then phase 3 would be a much bigger trial. So I have proved that my drug is safe and like also efficient for my deceased deceased population that I have, in hundreds. But before marketing it to the real world, we need to test it on a larger scale to make sure that the actual population is much higher. So we need to see whether at least like we need to mimic that population in our samples. So this phase 3 will mostly be a multi-center trial, like people from all the regions. If they are uh, planning to market it in India, it will be like a pan-Indian trial that will have like hundreds and thousands of people into the study, with basically more population, and then check for efficacy primarily. So this will be a confirmatory trial, and this will also go on as a long term trial. So once the phase 3 is done, the drug application will be submitted for approval. So and then the FDA or DCGI will review the results, and then it will be approved for marketing. So once it was put on market, once the drug is deemed to be eligible to be marketed, this were done in controlled environment, right? We have recruited subjects, we have monitored all the adverse reactions and all that. But once the drug goes onto the market, we cannot just leave it like that, right? I mean, you would have heard news about certain drugs being recalled every now and then, because they'll do conduct a long-term safety trials, post surveillance, post marketing surveillance studies, where they'll continue to monitor the safety and efficacy of the drug even after like being marketed. Even if you look at any of your drug that you usually take, if like look at it in the cover, you'll get the manufacturer name, and then there will also be instructions say if you have any adverse reactions, like contact this number, or like call this toll free number or whatever. So those now, when you have a reaction to a certain drug, that will be like recorded into the system, and then that will also be reviewed periodically. So that will be an ongoing process in the real world. So that will be like a phase 4 trial after the approval. Yeah. So if you have any questions, you can type it in the chat, or you can like keep it for the Q&A discussion. So yeah, moving forward, clinical, I also want to talk about the clinical trial management system, the process of how uh, the clinical trial happens. So first and foremost, we do the protocol development. We write the clinical protocol. uh, uh, Clinical protocol is like like the procedure and the literature behind this particular drug and this particular indication, and why we are doing what we are doing, how we are doing, and everything. And then once the protocol is approved, we'll do the site selection, like what are all the sites that we are going to use, and qualifying those sites and everything. And then we'll do site initiation. And then, like, if it's a multi-center trial, there will be more than one site, right? So we need to do all the site initiation. And then once we set up the protocol, once we set up the sites, if there are any necessary trainings required, if there are any audits required, everything will be solved. After that, we'll start patient recruitment. So these are like a milestones in any clinical trials: the first patient in and the last patient out. So first patient in, if, say I'm having 100 subjects. 100 subjects could come from any of the sites. 100 subjects could be of any gender, belong to any age category, depending on like what my inclusion and exclusion criteria are for my study. So and then study conduct, and then the last patients will also be part of the study. And then uh, treatment, like study happens. If I have multiple visits, multiple dosages, all that will happen. And then the last milestone will be last patient out. So what happens when the last patient has completed the trial? Imagine I have 10 subjects, I have say 4 visits happens every day, right, or every week. So it will take me 5 weeks to complete my trial for one patient, right? I have 10 patients, and each patient can start at any time. So say the first patient has started September 1st, the second patient would have started, like 11th September or whatever. So the first milestone will be when the first patient gets enrolled, and then the another milestone will be when the last patient gets their last treatment, and then moves out of the study. So once the last patient goes out, we have all the data that we need for this particular trial, right? So we will have all the data. The first step is to clean the data. We need to ensure the quality of the data, integrity of the data, make sure that are there any errors in the data, are there any ambiguity in the data. All that will be cleaned, queried, discussed with the site. The site, if there are any ambiguities, they'll go back to the site, talk to the site and see why is like this result is like that. Is there any specific reason, or is this an error? All those things they'll resolve, and then they'll lock the database so that there are no further changes being done to the existing database. And then we'll perform the analysis, right? Uh, so once we do the statistical analysis, we'll prepare the reports to be submitted to the regulatory authorities. So we call it a clinical study report. So this will have all the analysis, all the discussions that happened during the study, or all the information that is related to the study put together. We'll create a report and then submit it to the regulatory authority for submission. So we'll talk more about the analysis part moving forward. Yeah, so I have another question here. Let me just keep this for a minute. So uh, we create the protocol, and then we do the site initiation, and then we start patient recruitment, treatment and everything, and then we collect the data during this period. And then once all the data is collected, we'll do the data cleaning activities, and then we'll log the data, and then with the final data we'll perform all the analysis. And once the analysis is complete, we'll do the reporting, and then we will uh, submit it to the regulatories. Okay, so now I have my next question, which is we saw about the clinical trial management cycle, right? So what would be the role of statisticians here? Where do you think the statisticians would come in? I'll also post this link. Okay, you can start answering the polls if you can use the link in the chat. Okay, I see very few responses. You can just go to the link and then type the poll number. You'll get to see the poll. Okay, thank you. So all of you are saying throughout the trial cycle, I guess. Okay, once the trial is complete, we have few votes for beginning of the trial. We have few votes for analysis and reporting, but the majority is saying throughout the cycle. Okay, we'll see. So statisticians would be involved throughout the cycle. We'll talk more about at what phase, what kind of roles that would a statistician play. We'll talk about it in the later slides. So before that, I wanted to I wanted to talk about what are all the things that would be part of a clinical trial protocol, right? So protocol would have a title page, background information on the disease, and then background information on the the medication, and then the objective and purpose of the trial, because again we need to justify why we are doing what we are doing, and we need to evaluate whether the like the benefits outweigh the like impacts of the study. uh, so and then study design and endpoints. So what is going to be my study, how many doses am I going to have, what would be my dose levels, and what are all the endpoints that I have, what are the outcome measures that I have, and all that. And then study enrollment and withdrawal. Patients, how would they enroll, what would be the inclusion criteria, what would be the exclusion criteria, and all that. And then study procedures and schedule. uh, how frequently would the samples be collected, how frequently the tests would be done. And then efficacy assessment is how am I going to assess the efficacy of the drug, and then how am I going to assess the safety of the drug. So safety of the drug is basically your adverse events, and then investigations, blood investigations, and vital signs, physical examination, and all that. And then clinical monitoring. How do we monitor the data? How do we monitor the safety of the patients, etc.? And then statistical analysis. What kind of statistical analysis are we going to use? And then source documents. How do we control the documents? And then quality control of the whole trial. And then ethics, protection of human subjects, data handling, publication policy, study administration. Uh, all these will be part of clinical trial protocol. Everything would be completely and described to be able to conduct a clinical trial before we even begin the clinical trial. So in all of this, what are all the like parts that a statistician would be involved in while writing a protocol, right? So they would statistician would be part of the study design, from like sample size, the randomization design and everything. And then like the endpoint assessment, monitoring, statistical analysis, everything, like statistician would play a role while writing a clinical protocol, which would be completed even before the trial is like started. So what is the role of a biostatistician? So what do like people like me, people like my colleagues here do day in and day out, right? So from the clinical trial management system, we saw it. So there is a design phase, there is a study conduct phase, and then there is a study completion phase. During the study design, inputs to protocol, like the things that we discussed in the earlier slide, uh, inputs to protocol, defining the endpoints and everything. And then sample size calculation, randomization and blinding procedures, and then defining the endpoints for our study, and then defining the analysis population. So these are like real patients, real humans. It cannot go as if like exactly as we planned, right? So what if a patient missed a dose? What if a patient like left the study before completing all the scheduled visits? So we need to define what would constitute as a acceptable analysis population for different kinds of analysis. We would try to come up with like criteria to include the patients into the analysis. And another thing is, like, we are this is like, even if we have partial information, we should be able to use that information in our analysis, because that patient has also taken the drug, and we are also responsible to like ensuring their safety and everything. So we define analysis population for our study, and then formulate hypothesis, and then defining statistical methods and analysis that would be appropriate for our own clinical study. So all these would be done even before the patient recruitment happens, right? And then study conduct. So once the patients are like being enrolled and everything, uh, we'll do data monitoring. So data monitoring is usually done by clinical data management team. but still, like there are strategies that we discuss, there are endpoints, and we do risk based checks and everything, which also requires a statistician's input. And then data plausibility checks, and then blinded data review. If you're doing a blinded study, we'll have like we need to review the data on a periodical basis to ensure the safety of the drug, right? So even if I am having a set of subjects with my drug and a set of subjects with placebo, I need to ensure safety of all patients. So we do review and report data in a periodic manner, but also those will be like a blinded data. And then quality control to evaluate the integrity of the database. So all these we'll be doing during the study conduct phase. And then once the study is complete, during the completion phase, this is where the huge chunk of works come in. After, like the designing phase, we'll have all the data available. So we'll do the final analysis, and then we'll do the statistical interpretation of the analysis, and then we'll support the clinical study report process to ensure that all the endpoints are interpreted correctly and statistically meaningful. Yeah, so this is what we do as biostatisticians in clinical trials. So moving forward, I just wanted to give you a few examples of what we do with respect to study design. So study design should be explicitly defined, all the visits, how many samples I'm going to collect, how many doses I'm going to give, what are all the samples I'm going to collect. Everything needs to be explicitly defined, and only whatever is being defined in the protocol will only be followed throughout the study. And then to calculate sample size, there are different designs for depending on our study, like the kind of groups we have. Do I have parallel group design, which means like different population taking different kinds of drugs, and then crossover design. Crossover design is where uh, if if I am testing two drugs, drug A and drug B, the same set of subjects will have, we'll have two phase. So drug A and B, in the phase one group will take A, one group will take B. And then in the next phase, it will be reversed. The subjects who took A will be given drug B, and then the subjects who took B will take drug A. So this will give us more uh, uh, results and interaction results. Like, see, I'm comparing, like if I'm doing a parallel design, if I'm taking drug A and, like my colleague is taking drug B, I'm comparing my results with another person's results, right? So this is also to valid and relevant to certain extent, but when I'm doing a crossover design, I can compare my results with the other person, and also I can compare, and I can also compare, like my results with like my results itself with the like from for the two different drugs. I can compare my results. So this would also like we also need very less subjects recruited because to meet our sample size. But the drawback would be that we need more time. Like if I recruit different subjects for different groups, I can complete it in a certain time. But if I'm doing if my study is like probably 5 weeks treatment, I need only 5 weeks for each patient to be completed. But if I'm doing a crossover design, I need 5 weeks, and then probably 2-3 weeks for short period, and then again another 5 weeks for one patient to complete the study. So the trial time duration gets long. So uh, all these impacts how we calculate the sample size. And then hypothesis testing. What would be our trial objective? Do we want to like prove equality, non-inferiority, superiority, or equivalence? So what do we want to prove with our trial, with our drug, to the with respect to the comparator? Yeah. So moving forward, I have an example study design. Yeah, so I have active controlled randomized trial. So that proposes to assess the effectiveness of drug A in reducing pain. So a previous study showed that drug A can reduce pain score by 5 points from baseline to week 24 with a standard deviation of 1.195. A clinically important difference of 0.5 as compared to active drug is also considered to be acceptable. Consider a dropout rate of 100. So how do I calculate the sample size for this? So I'll set my level of significance at 5%. I'll have the power of at least 80. And then this will be a two-sided test. So what do I have here? I have, so baseline is um, any last measurement before start taking the drug. So usually in pain related studies or any other studies are one of the major outcomes would be to see the change, after taking the drug, right? So how do I define that change? So I'm doing a trial for pain. So obviously I'll measure the pain before start taking the drug, right? So I'll have the measurement of the pain score before I start taking the drug, and then once I start taking the drug, I'll continue measuring the pain score on the same scale so that I can compare at any point how much it has changed from baseline, because baseline results are the ones without the drug. So after taking the drugs, whether it's like decreasing or increasing, or on what scale it is increasing and decreasing and all that. So even a 0.5 difference of compared to active drug is considered to be acceptable. So mu1 would be the mean change in pain score from baseline to 24 week 24 in drug A, and then mu2 would be the same in active drug, which is 5 in our drug A and then 4.5 in active drug, because we see that even a 0.5 change is acceptable. So our significant difference would be 0.5 and the SD would be 1.195 based on the literature. Yeah. So based on the formula, the sample size required per group is 90 based on the sample size formula. So we have two groups, right? So one group, each group would have 90 patients. So I would need 180 patients. And this is a real time study. So there would be a lot of reasons, why a patient could stop or terminate from the trial. So we would, usually consider a 10% dropout rate, considering the trial design and how long the trial is and everything. So for this study, I'm considering 10% sample size, and the sample size required would be 200, where I'll have 100 in each arm. So the protocol description would be like, a sample size of 180 subjects, 90 in each arm, is sufficient to detect clinically important difference of 0.5 between groups in reducing pain, assuming the standard deviation of 1.195, using a two-tailed t-test of difference between means with 80% power and a 5% level of significance. And when we consider a 10% dropout rate, the sample size required would be 200. This is an example of how we calculate sample size in real time. So we also saw about the objective of the trial. So what do we want to prove? There are superiority trials, non-inferiority trials, and like equivalence trials and everything. So say I am bringing a new drug to the market. So what do I want to prove? Say if I'm curing something that has not been cured before, say if it's a rare disease or a cancer or something, I can, like cancer trials have different set of rules. They work with different kind of objectives, because we don't have a cure for cancers yet. So you can only see how my drug improves the quality of the patient, or how my drug is, like helping, like retaining the like disease progression and everything. So I'm not talking more about oncology. We'll probably touch upon it in some time, but like superiority trial is for any other drug where we'll see whether the my investigational drug, whether it is better than the pre-existing control trial. And then non-inferiority trial is, I'm saying I have this drug is already in the market, and then I'm bringing a new drug. I'm just saying it's not like worse than the actual trial, I mean the active drug, which means that whether it could be better or whether it could be equivalent, but it's not like worse than the pre-existing drug. And then equivalence trial is, these two are like similar. uh, why do I want to say like it's not better but it's equal? Sometimes like I have, like the paracetamol example, I have paracetamol in tablets, but I want to make it into a syrup so that like it's easier to give it to kids or even for like adult population. So what I'm doing is I'm just doing a trial with syrup, and then comparing the results with like with the capsule, and I'm saying the results are like equivalent, and each, I mean both can be used interchangeably. So survival analysis uh, survival analysis is the major part of oncology cancer trials. So what we do is mostly we'll have the time to event data. So we'll have subjects, and then we'll have a time duration defined for our study. So usually we need event in oncology trials. So event would be death, right? So say I'm doing the trial for 3 years, I'm following up for 3 years. Some subjects may have died in those 3 years, and then some would have been alive during the end of the study. At the end of 3 years, some subjects may have been alive, but some subjects may also have been left the study, right? So so the patients who have event, I know for sure that they had the event and then their duration to the event. But for the rest of them, will do something called censoring. So we know up to this point that this person hasn't died, right? After the study, the subject may or may not have died, but like we don't have that information. And like similarly, this participant may or may not be alive, we don't know that. So we will use the information that we know for sure, which is we know that this person was there until until the end of the study. So these are some of the endpoints that we use, like during the start of the treatment. And then responses, like whether the uh, disease is the tumor like is going down and the patient is getting better. And then progression is when the disease is getting worse. And then death. So time to response would be from the beginning of the treatment to when they have a positive response. Then time to progression would be from the treatment of trial to when they start getting worse and the disease is getting progressed. So survival time is from treatment start to the event, which is death. So these are some of the endpoints that we use in survival analysis. Overall survival, this is like similar to what we discussed. Like from this, what the different times we have, like different definitions and endpoints and what you want to see. Considering the time, like I'm just not getting into detail with this, but survival analysis is a huge topic in itself, which is also like even more interesting. So feel free to go through this like when you have time. I also want to touch upon this topic, which is my favorite topic. Right now I work with biomarkers. So to start with, what is a biomarker? Biomarker is just a biological characteristics of an organism. There are any kind of measurement, any kind of biological characteristics can be called as a biomarker. But the more we get into in clinical space, we talk about the gene expression within cells and proteins in blood, proteins in bone, and everything we call as biomarkers. And we collect these biomarker samples, analyze them, and then uh, uh, to take a step into understanding the human body even more like in depth, right? So why biomarkers are important in clinical research in the past 10-15 years, especially in cancer trials. There were a lot of changes that has been happened because we were able to understand the mutations. We were able to understand what a human body goes through when there's a cancer. And when we get to understand it on a gene level, we were able to bring more advanced techniques and more targeted treatments that would help the patients with that certain disease. So biomarkers are becoming very important in clinical research, especially in understanding the disease and understanding the characteristics of the disease, and also the human population that we are having. So I just want to show you a few of the cool graphs that we do to understand. So these are all the sequencing data measurements we have for each of the patients. So if they have mutations, we'll just code it differently. And uh, this is how we do biomarker analysis as an example. So another aspect of biomarker research is what do you see here? You can type it in the chat if if you have any thoughts on this. What do you see here? It's the same it's the same cloth, it's the same bowl, but it doesn't mean that it fits everyone, right? It may fit somebody, but it may not fit everybody. So the question here is, so does one size fit everyone? That's the question that we are asking right now. Sorry, is someone, can you all make sure that you're muted? Yeah, thank you. So yeah, so the question that we are asking is, does one size fit all? In clinical research, in the techniques and technical advancements that we've had over the years, the marvelous innovations that have happened, we are at the point where we're thinking we have treatments for most of the diseases, but does the treatment that we have, does it work for everyone the same way, right? One treatment could work for somebody and one may not work for the same treatment may not work for someone else. So we are getting into the world of biomarkers with respect to precision medicine. So yeah, we have people of different groups with having different kinds of characteristics all over the world, but having the same treatment may work differently for different people. Some people it may be effective, some people it may be effective but may have stronger side effects also. So some people it may not work at all. So what do? We, based on whatever we know, we design studies that helps each of the population, each of the groups separately, based on their own targeted cells and targeted therapy, which would uh, be be the precision medicine. But the key derivative for this would be the biomarker results that we have from all the samples that we have collected, and and to understand each of the population and their differences to get better target healthcare. Yeah, that's it about my presentation. I'm sorry I took up a lot of time from the question and answer. But yeah, if you have any questions, you can unmute yourself and ask.
So we are in our Q&A session. So if anybody has questions, you may either type in the chat box or you can just unmute yourself and ask. And just to engage with each other, I would urge everyone to just share their application in the chat box. It will help all of you to just to get to know each other. There is one question, the difference between NDA and IND.
NDA is new drug application. What is IND? Shakti, right? Do you know what is IND? Okay. So, investigational new drug is the drug that I am testing, right? See, I'm having paracetamol. I'm testing paracetamol, or I'm using, like creating a new drug for migraine. That drug would be called IND, investigational new drug. But the NDA is new drug application. So, with this drug, what am I planning to do? What is the objective I have? What is the proposal protocol that I have to the regulatory authority? I need to submit all the literature, all the like protocol plans and everything to the FDA or DCGI to get it approved so that I can start the trial. So that application is called NDA, right? So NDA is the application, and then IND is the drug that I'm using for the application. Thank you, Dharni. Any other questions?
So if there are no further questions, I would like to thank the speaker for accepting our invitation and for this enriching talk. We do also look forward to you joining Caucus soon. So
Yeah.
thank you very much, and thank you all for coming. So if you have any questions, you also have the email. You can always shoot an email.
Yeah, sure. Feel free to reach out if you have any questions, anything. Yeah. Thank you, Sangeetha, for providing me the opportunity and inviting me here. Like I appreciate it. Thank you so much.
My
Hope it was
pleasure.
useful for everyone here. Yeah. Thank you.
It was really helpful. Thank you.
Thank you.
So, okay, so I'll end this call.